Hello, hi, a very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So thank you so much for joining my session this evening. So we are here talking about lecture 4 of biomolecules. So I hope you guys are doing well and you guys had a good weekend. Yes? Okay, so we were talking about the portion of biomolecules and biomacromolecules, the acid soluble fraction, the acid insoluble fraction, and we're talking about the structure of amino acids, the structure of your nucleotides, and then we went on to talk about the structure of proteins. We also spoke about the structure of lipids. So we were talking about the structure of proteins, so we'll move ahead and finish off the structure of proteins. Yes, ma'am, a very good evening. So thank you so much. So you're my first student here to watch and wish me, so thank you so much. So please don't forget to like, share and subscribe. Yeah, so let's move forward. So let's finish off biomolecules. Okay, so here we are. So to give you a recap, we started with the structure of biomacromolecules, right? So before that, we are talking about your metabolites, your primary metabolites, your secondary metabolites, and so on. So we are here talking about your biomacromolecules. So biomacromolecules, this includes your proteins, your nucleic acids and your polysaccharides. So these three make up your biomacromolecules. So talking further about your proteins are polypeptides. So they are made up of amino acids which are connected together by your peptide bonds. So that is your polypeptides. But proteins are heteropolymers, meaning they are made up of many number of amino acids or many types of amino acids. So when I write like this, amino acid 1, amino acid 2, amino acid 3. So the three of them are different amino acids. So this can be your glycine, this can be your alanine, this can be your serine. So all of them are different amino acids. So it is a heteropolymer. It is not a homopolymer. Hetero is different. So it is made up of different entities here. So different types of amino acid that makes up your protein. So if you see, I never added an amino acid like this. I never wrote amino acid 1, 2, 3, but instead I wrote it from left to right, not from right to left, right? So always remember, we add a new amino acid only towards the right hand side and not towards the left hand side. So recollecting the structure of amino acids, so we have an alpha carbon, so we have a hydrogen bond, so we have a carboxyl group, so we have an amino group, and we have an R group. Right, so thus the right hand end of the amino acid of the protein is called as the C terminal end or the carboxyl terminal. So this is the C terminal or the carboxyl terminal of a polypeptide, and this is the N terminal or the amino terminal of a polypeptide. Fine. So far clear? Any doubt? And this is the primary structure of protein. So when you talk about the structure of proteins, the protein structure can take up to 4 stages. So you have the primary structure, you have the secondary structure, the tertiary structure and the quaternary structure. The primary structure is a linear sequence of amino acids. So this is your primary structure, or you can say the linear chain. So this is the primary structure, or this is the linear chain of amino acids. Right, so this is how it is written. So moving further, we go to the secondary structure. The secondary structure consists of alpha helix. So these are your helix and these are joined by pleats. So we call them as beta pleats and alpha helix. So this makes up your secondary structure, right? And in your tertiary structure, you have what is called as subunits. So if you remember, we read about hemoglobin. So hemoglobin has 4 subunits. So it has 2 alpha subunits and it has 2 beta subunits, right? So each subunit is comprised of a tertiary structure. So now talking about your quaternary structure. So this is made up of more than 1 subunit. So that is your quaternary structure. Alright, clear? So this is the structure of your amino acids. Any doubts so far? The structure of the amino acid is clear? Yes? Okay, so can we move forward now? Okay, so now I will show you the pictures of the corresponding things that we discussed so far. So we were here talking about, okay, so polysaccharides have come. So okay, so this is about your proteins. Okay, so I hope you have understood the concept about your proteins properly, right? So the bonds between the proteins are your peptide bonds. So let me tell you, so when I wrote like this, amino acid 1, amino acid 2 and amino acid 3. So how are these two amino acids connected? They are connected by what is called as your peptide bonds. Right? So how do I write an amino acid? Like this. So the next amino acid should be like this. So this is my amino acid 1 and this is my amino acid 2. Okay. So now I have to join these two. So these two are joined by the elimination of water molecules. So when I eliminate water molecules, so what will be remaining will be your NH and CO here. So from here only CO will be remaining, from here only NH will be remaining. So now I write the entire structure like this. So this bond is a peptide bond. This bond is a peptide bond. Alright. So peptide bond is a type of dehydration reaction where water is eliminated. So water is eliminated and it forms a peptide bond. So with this we are done with the protein part of the biomacromolecules. So now we'll move into the polysaccharide portion. Alright. So acid insoluble pellets have your polysaccharides and they are nothing but your carbohydrates. Alright. So polysaccharides are long chains of sugars, just like proteins are long chains of amino acids. So these are long chains of sugars. Alright. So these are monosaccharides which are made up of, which makes up your chain. So it is a long chain. So polysaccharide is a huge molecule. It is made up of individual units. We call something as a train. And if you have only two compartments and each box is a compartment, right? So a train is made up of many compartments. So that is how your polysaccharide is. So your polysaccharide is made up of many individual units, like a train is made up of many compartments, and you have an engine, you have a compartment, etc. Right? So in a polysaccharide, the individual units are called as monosaccharides, that is single sugar. Poly is many. Right? So they are made up of monosaccharides as building blocks, just like amino acids are the building blocks in your proteins. So here monosaccharides are the building blocks. Alright. So cellulose, which is a polymeric polysaccharide, so it is a long chain and the individual monosaccharides are glucose. So cellulose, so you know plant cell wall is made up of cellulose. So cellulose is made up of cellulose is a polysaccharide. So poly means many, saccharide means sugar. So this is many sugar, right? So it is made up of many molecules of glucose and each glucose molecule is a monosaccharide. Fine. Each glucose is a monosaccharide here. So cellulose is a homopolymer in the sense it is made up of the same monosaccharides repeatedly. So everything is made up of the same glucose, unlike the protein which is a heteropolymer. So protein is made up of different amino acids, but cellulose is made up of the same monosaccharide, that is glucose. So cellulose is your homopolymer. Alright, so starch, which is a variant of your glucose. So you can say plants by photosynthesis they produce glucose and they store it in the form of sucrose, and in animals they store it in the form of glycogen, right? So starch, which is a variant, and it is also stored, glucose is also stored as starch and it is a storehouse of energy in your plant tissues. So in animals it is stored as glycogen. So your starch is a polysaccharide, which is a helical polysaccharide. In between these helix, iodine is stored. So your starch is also polysaccharide, but your starch has a helical structure and in between the helix your iodine is stored. Alright, so this is the structure of your starch, though it is made up of the same monosaccharide, and you have something called as inulin. So inulin is a homopolymer of fructose. It is made up of repeating units of fructose. Alright, clear? So we go further. So in a polysaccharide, the right hand is called the reducing end, the left end is the non-reducing end. So starch forms helical secondary structure. It can hold iodine molecule. So that is why to test the presence of starch for photosynthesis we use iodine. So that is the only reason we use iodine to test for the presence of starch. Alright. Okay, so if you look at, so we have the structure of glucose here. So we have glucose 1, 2, 3, 4. So probably it's forming a chain, it's forming your polysaccharide, that is cellulose. So if you see in glucose, this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon, right? So first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. So glucose is a 6 carbon molecule, right? Yeah. But when you see again only on the right hand side, I am adding the new molecule. So when I say write glucose, I write it only on the left side. I never go and write it this side. So the right hand end is called as the reducing end. So this can reduce to accommodate an incoming molecule, whereas this cannot reduce to accommodate an incoming molecule. Alright, so if you see here in the first position, so it forms a glycosidic bond. So again by the elimination of water molecules, it forms a bond called as the glycosidic bond. Fine. So the glycosidic bond is formed by the first carbon of the first glucose and the fourth carbon of the corresponding glucose. So you can say alpha 1, 4 glycosidic bond, because this is alpha type linkage. So this is the bond between the corresponding glucose. So this is the reducing end, this is the non-reducing end. In case of amino acids, it's the carboxyl end and it is the N-terminal end. Alright. Yeah, so this is how the structure of starch is. So in between the secondary helix, it can take up or it can hold iodine. Alright. So there are certain polysaccharides like your cellulose which make up your plant cell wall. So paper is made from paper pulp and cotton fibers are cellulosic sometimes. So sometimes you can have a complex version as well. So sometimes you read pure biology, sometimes you read with conjugation with chemistry like your biomolecules, right? So there are certain molecules which are pure polysaccharides. There are certain molecules which are called as complex polysaccharides. So complex polysaccharides like your paper pulp and your cotton fiber, which is a bit cellulosic. Alright, so the complex polysaccharides are made up of building blocks, amino sugars and chemically modified sugars. Yeah, they are complex, so from the name you should understand it has a bit of complexity. So they have modified sugars, that is glucosamine and acetyl galactosamine. Right? For example, when I say I have a lot of glucose molecules, say for example I have repeated cellulose molecules, these cellulose can be joined by your N-acetyl glycosamine. N-acetyl galactosamine or glycosamine, they are also modified sugars. They connect two polysaccharides together, thus forming your complex polysaccharide. Alright? Yes? So arthropods have exoskeleton. So if you have read cockroach, yes, you should remember. So your arthropods like your cockroach. So if you see they have a brown skeleton, brown surface, so that is your exoskeleton, and that is because of a complex polysaccharide, that is your chitin. So chitin is homopolymers, they are made up of repeated monosaccharides. Alright. Yeah, this is how your N-acetyl glucosamine is. So it links two glucose molecules together. So this is your glucamine. So this is N-acetyl glucosamine. Alright. Yes. And this brown layer is your chitinous exoskeleton. That is your homopolymer. Right. So then we talk about your nucleic acids. They are found in the acid insoluble fraction. So they are made up of nucleotides. So your polysaccharides, so PS is polysaccharides, polypeptides are your proteins plus your nucleic acids. They are true macromolecular fraction of any living tissue, right? So they form the building blocks of your nucleic acids, that is your nucleotides. So we know the structure of nucleotides, so it has one nucleoside and a phosphate. So the nucleoside is made up of a nitrogenous base and a sugar, right? So yeah. So this is how it is. So we know how the sugar varies in your RNA and in your DNA because I've written the entire tabular column of nucleotides in the last class. So I hope you guys remember, right? So here we are talking about the structure of proteins. So proteins are heteropolymers. They contain a string of amino acids. So primary structure is just a sequence of amino acids. It gives you the positional information in a protein as to which amino acid is present where. For example, serine is the 4th amino acid, glycine is the 2nd amino acid. That is the information that it gives. Alright, so the left end is the first amino acid, so that is the N terminal. The right end is the incoming last amino acid, and that is the C terminal. So the secondary structure has helix and beta pleats. The tertiary structure is necessary for many biological activities, that is, it forms your subunits. So this is your primary structure where it gives you a sequence of amino acids. You have your secondary structure which has a helix, right? And then you have your tertiary structure, so it has your beta helix. It has many beta helix put together and many subunits put together. You have the quaternary structure. All right. So some proteins have assembly of more than one polypeptide or subunit, so that is your quaternary structure. For example, hemoglobin. So hemoglobin is present inside your RBC. It has 4 subunits, so 2 of which are identical to each other. So it has 2 alpha and 2 beta subunits, right? Yeah, this is how is your hemoglobin. So you have 2 beta and you have 2 alpha subunits, all right? Yeah, to be very clear, so we have differentiated all the 4 subunits in 4 different colors, okay? Right, so we are done with your major portion of your acid insoluble fraction. So we are done talking about your proteins, we are done talking about your polysaccharides. So we are done talking about your nucleic acids as well, but once again I will explain the structure of DNA, though we have already seen it in the molecular basis of inheritance. So I will revise it once again so that we'll be done with this portion. So very soon we'll be talking about the enzymes. So enzymes are the most important part of your biomolecules. So I'll come with very interesting topics on enzymes. So I will teach you with a lot of beautiful examples and GIFs. So thank you so much for watching my lecture today. So please do like, share and subscribe. So I will meet you on Wednesday at 4:30. So until then, this is me signing off. Take care and bye. Yes, bye. Thank you.